looking at mood disorders to set the tone for the rest of the symposium, because this is such an important topic in psychiatry, namely personalized medicine. So this is the holy grail in psychiatry right now. There are really two aspects of personalized medicine in psychiatry. The first portion of personalized medicine relates to disease vulnerability, and that is, can you identify individuals who are at risk for a mood disorder, in this case, or an anxiety disorder, when Dr. Pirna gives his presentation. Because if you could identify an at-risk group, you could intercede before they ever have a full-blown syndrome. And this is true of schizophrenia, bipolar disorder, and depression. And in order to understand this, one of the themes of the symposium is summarized on this slide, which is, how does genetic vulnerability result in cellular changes that result in circuit changes that result in the phenotypes of these psychiatric disorders? And each of the speakers will address different aspects of this. Dr. House will talk about a particular gene, the FKBP5 gene, and how it interacts with epigenetic mechanisms to increase disease vulnerability. Dr. Pirna will be talking about personalized medicine and anxiety disorders. Dr. Schlepfer will focus on brain circuitry, and in particular, the role of deep brain stimulation and other non-pharmacological somatic treatments. So let's use as an example bipolar disorder. This is a well-known slide depicting the course of bipolar disorder. We know that the genetic risk for bipolar disorder is very high. 2/3 of the risk to develop this disorder is genetic. It's the most genetically laden of all of the major psychiatric disorders. And 1/3 is environmental. And one of the environmental factors I talked about yesterday was early life trauma. But the fact of the matter is, we know very little about the course of this illness from a biological point of view. We know that most bipolar disorder starts with early life depression, pre-pubertally, and then over time patients go from having depressive episodes to having manic episodes, and the more mature form of the disorder is what we see here near the end of this course. But we know very little about this disease. So again, what we want to do is we want to be able to identify individuals who are at risk so that we can develop a preventative strategy, a vaccine, for example, to prevent drug abuse, to prevent bipolar disorder, to prevent the development of PTSD, certainly to be able to intercede early and hopefully before the patients have the clinical manifestations of the illness. So I want to spend a fair amount of time talking about genetics in my presentation. And all of you know the human genome was sequenced by the Human Genome Project as well as Craig Ventnor, and there were these articles that came out suggesting that this was the answer to everything, that we knew the future of medicine was all in our hands. We had the secret of life, as you can see here, and this was going to tell you everything you needed to know about disease. And Jim Watson, who discovered the double helix, he paraphrased Shakespeare. He said, now, we used to think our fate was in our stars, and now we know it's in our genes. And here are the psychiatric disorders, and you can see the most genetically driven of all of the complex diseases, more than diabetes, more than cancer, bipolar disorder, schizophrenia, depression. All of our illnesses are very genetically driven. And we know this from old studies that show that the concordance rate for bipolar disorder in monozygotic twins is higher than in dizygotic twins in every single study that's ever been done. And this is ironclad evidence that a large part of the risk for bipolar disorder is genetic. In major depression, it's about 35 to 40%, as you can see in this meta-analysis that Pat Sullivan published several years ago. One of the problems we have in psychiatry is that our nosology lumps together diseases with very different biological etiologies. So this is all the mood disorders across the life cycle, and everybody knows that early onset depression, which is often bipolar disorder, is biologically very distinct than postpartum depression and very distinct from late life onset, which is more of a vascular depression. Yet all of these things are called major depressive disorder. And that's one of the reasons why we've had so much trouble in treatment trials. So early studies looked at linkage, and this was before the sequencing of the genome. It didn't tell us anything about individual genes, but it told us that there were regions of the chromosome that appeared to be overrepresented in patients and families with bipolar disorder. And these were crude studies. They identified certain locations on various chromosomes, but they were good for what we did then, but they didn't identify any genes. In the meantime, we were trying to understand the biology of these disorders, and we focused largely on serotonin, norepinephrine, and dopamine. And we had this idea that each of these neurotransmitters have a different role, and that together they contributed to the biology of mood disorders. And of course, major depression in particular. But in a given patient, we could never know with any easy laboratory test, did they have a dopamine problem, did they have a serotonin problem, did they have a norepinephrine problem? And let's remember that depression is pretty complicated. As you can see, it involves a large number of brain regions, as Dr. Schlepper could tell you, which underlie the different symptoms of depression, ranging from alterations in cognition and executive function, to alterations in sleep, to alterations in fear responsiveness, to an inability to experience pleasure. And these are multiple neurotransmitter systems. So as far as the serotonin system, from an individual point of view, it is true that if you look at PET or SPECT studies, you can identify depressed patients, as in one of our studies, that have low serotonin transporter binding in the midbrain. But the fact of the matter is, in spite of being able to replicate this in suicidal patients, our group, John Mann's group at Columbia, it's not been a good predictor of treatment response. And in fact, we have a study just completed in never before treated depressed patients showing that serotonin transporter occupancy does not predict treatment response to escitalopram or duloxetine. No relationship whatsoever, a study of 400 patients, which we're just finishing writing up now. So that you were just as likely to respond to escitalopram if you had 20% occupancy than if you had 90% occupancy. So we have to rethink the serotonin hypothesis of depression. Dopamine, certainly important in Parkinson's disease. There's evidence, as you see here, of a decrease in dopamine transporter binding in the both in the striatum and in the nigra. This is the striatum in depressed patients compared to controls. Age-related decline, as you see here, but again, not predictive of response, and evidence that norepinephrine may be involved from postmortem studies. But again, these drugs do not in any way result in a high rate of remission in depressed patients. Remember, the STAR-D trial, only 28% of patients treated with citalopram up to 40 milligrams a day achieved remission. That means that 3/4 of the patients with major depression did not achieve remission with an adequate dose of SSRIs. So what's the problem here? And I think we have to go back to the genetics. And let me go over this for a minute. What you're looking at here is the classic Watson-Crick model of DNA transcription to messenger RNA. Messenger RNA leaves the nucleus and then is translated on the ribosome translated into the protein that the DNA codes for. And the genome has been sequenced, and there it is. Isn't that exciting? It reads like a novel. And 99% of the genome is the same among everyone in this room, with the exception of these little variations that are called SNPs. And there's several million of these SNPs, and they're partly responsible for why we don't all look alike, we don't all sound alike, we don't all feel alike. So for those of you who are not genetics aficionados, just to remind you that here's the triplet code coding for amino acids. Here is a SNP, an amino acid, a nucleotide substitution, a mutation, but it doesn't change this triplet code which amino acid is coded for. So that would be called a non-functional SNP. But in this case, you can see this SNP is functional, so that the amino acid coded for, instead of an arginine, is a lysine, and that results in a change in the tertiary structure of the protein, which makes it a better receptor or a worse enzyme, etc. And it's these functional SNPs we're interested in. So the field was largely excited by this report by Alvin Caspi at the Institute of Psychiatry in London, where he looked at the genetic polymorphisms of the serotonin transporter, and there are two in the promoter region. There's a long arm and a short arm allele, and it turns out this is a functional SNP, and if you have the short arm, either as a heterozygote or homozygote, you actually express fewer serotonin transporters in the midbrain. And in this famous paper published in Science now more than a decade ago, he showed that if you have this S allele, either as a heterozygote or homozygote, and you're exposed to early life trauma in the form of child abuse and neglect, you have a markedly increased risk for depression and, it turns out, suicide. Whereas if you have the long arm homozygote, you're protected from the effects of child abuse and neglect. This is an example of personalized medicine. I talked about this yesterday. The other system that's gotten a lot of attention, of course, is the pituitary adrenal axis because of the role of stress. And we know that the CRH system is hyperactive in depressed patients. I mentioned this in my presentation yesterday. So we did this study looking at depressed patients. We're looking at a large population of patients, 484 African Americans, and we measured their child abuse and we measured their depression severity, and you see the relationship between child abuse and depression severity. And we identified, as I mentioned yesterday, these three variants of the CRH1 receptor, which mediated the depressogenic effects of early life trauma. Here are the protective SNPs, and here are the vulnerability SNPs. But then, with Elizabeth Binder collaborating with us at the time, we were able to begin to look at another gene. So what this slide shows is that early life trauma coupled with adult trauma increases the risk for post-traumatic stress disorder. This is the post-traumatic stress disorder symptom severity scale. And the more child abuse you have and the more adult trauma you have, the more likely you'll develop this. And here's the FKBP5 gene with the three SNPs that were uncovered to mediate the likelihood that this gene contributes by interacting with child abuse to increase the risk for PTSD. And we're able to show this in more than one study, and the data, the more child abuse you experience, the bigger the effect. I'm sure Dr. Hausch will be talking about this. Now, the other field that has been exploding, of course, is epigenetics. And what that is, of course, is the notion that you can change gene regulation without changing the sequence of the gene by post-translational modifications. And all of you are aware of the fact that by process of either demethylation or deacetylation, you can change the gene expression by effects on the histone proteins that are so closely linked to the double helix. And this process has been well described, and it has a role in cancer, it has a role in diabetes and in other disorders. And so with Torsten Klengel, we were able to show that the FKBP5 risk allele is actually a risk allele due to the effect of demethylation as a function of child abuse severity, and in that risk allele alone. And Dr. House will follow this with a discussion of how you can take this work and then translate it into potential novel therapeutic areas. So let's get back to, I want to spend a few minutes being critical of GWAS studies. So GWAS has been the approach that has been suggested to provide us with the way that we could discover why we haven't in the past been able to uncover genes for risk for major psychiatric disorders. And what you have to understand about GWAS studies are they're completely ahypothetical. There's no hypothesis being tested. You simply take a large number of subjects with and without the disease, you compare the distribution of the various SNPs, and then you determine whether the patients with the disease have a preponderance of one genetic variant compared to another. And this paper was published last year, which suggested that this is a phenomenal finding. So they looked at 24,000 patients and controls together, and they reported identifying 56 genome-wide significant SNPs in 5 different regions. So this is the Manhattan plot. This is each of the non-sex chromosomes, so 22 chromosomes. This is a statistically significant line, as you see here, which has arbitrarily been placed at 10 to the -7. And you see that for each of these chromosomes, there are a few SNPs here that occur above the line. And then if you blow this up, you can see the actual raw data so that you can see there are some findings here. And if you look at the sample, you can also tell that this would be no effect, that with the exception of a few countries, almost all of the countries that contributed samples here showed the same pattern in these 2 particular chromosomes that I showed you on the previous slide. So that seems pretty exciting. And here's a big, this is from the schizophrenia GWAS. I want to compare it. You can see this big finding here. So that sounds pretty exciting. But I want to give you a little dose of reality. The reason these findings are so significant is because the numbers are so large. If you're studying thousands of patients, you can see differences, statistical differences with a very large number of patients, very small differences that are statistically significant. But what you're interested in is in a given person, is it predictive of disease vulnerability? That's what you want to know. So I just want to show you these findings. These are the 2 biggest findings in yellow from the schizophrenia GWAS study, and the bipolar study is similar. These are the frequencies of these 2 genes in the control population and in the disease population. And I want you to see the frequency differences. So this was 10 to the -8, this p-value, and what you're looking at is 31.9% versus 30.1%. And the second one was 37.7% versus 35.6%. And here are the odds ratios. Look how low they are. 1.07, 1.07. So this doesn't give you any power to predict in a given individual This may provide you with a lead about the biology, but it doesn't provide you with any information that's going to help you in terms of personalized medicine. So why has it been so difficult for us to find genes that are responsible for the major psychiatric disorders, if indeed these disorders are in fact so genetically laden? And let's get back to the fundamentals about genes, which is if you think that we are the most sophisticated species, you would think we would have the most number of genes. But look, what I'm showing you here is that we don't. We have slightly more genes than the sea worm and the fruit fly, and here's seaweed has more genes than we have. And remember, psychiatric disorders are unique to our species. So what's responsible for complexity? Because if we understand that, we probably have a clue as to what's responsible for psychopathology. And the answer turns out to be not genes, but RNA, which sounds a little counterintuitive. This is what we were all taught when we were in high school and in medical school. The DNA codes for messenger RNA and messenger RNA codes for protein. But it turns out that the vast majority of DNA codes for non-coding RNAs, microRNAs, that do not code for proteins. That's a major finding in the last decade. What is going on here? Well, if you plot the proportion of non-coding DNA with developmental complexity, here's humans up here. It plots exactly correctly in terms of the association. The more microRNAs, the more non-coding DNA, the higher the complexity. In fact, only 1.2% of the genome actually codes for enzymes, receptors, structural proteins. That means that 98.8% of the genome doesn't code for proteins, that they code for microRNAs. So we have a whole new view of the human genome, and that view is that there are small islands of conventional protein coding genes that are living in a sea of microRNAs that regulate both transcription and translation. And we've now come to understand that these regulate splicing, so there are pro-hormones like beta-endorphin, pro-melanocortin, which codes for beta-endorphin, which codes for metenkephalin. What about the differential splicing? A single microRNA can target multiple genes in a given pathway. We know that they have a major role in development, and we know that many of our disorders are disorders of development, and we've already seen reports from our group and others of abnormalities of microRNAs in psychiatric disorders. So in the last few minutes, I want to talk about the other half of personalized medicine in psychiatry, not disease vulnerability, but prediction and treatment response. You see patients in your office and you're trying to figure out what drug should I prescribe for them, for bipolar disorder or for unipolar depression. We all think we know, we think we have a good idea of how to predict response, but we're fooling ourselves. We don't have good data. So in 2004, I published this report in the American Journal of Psychiatry, which is the least cited of any paper I ever wrote. I think it was probably before its time, but it was looking at the prediction of response to milnacipran as a function of the norepinephrine transporter gene in a Japanese population. You can tell that I'm not Japanese from the author list. But we were able to identify a SNP that was associated with a poorer response to milnacipran of the norepinephrine transporter, and one that was associated with a slower response. And then Elizabeth and I looked at the STAR-D data. We were able to show that there were components of the CRF system that influenced treatment response to citalopram. We published this a couple of years ago. Elizabeth went on to look at FKBP5, her favorite gene, and was able to report that it was associated, that variations in this gene were associated with a good or a poor response to SSRIs, to tricyclics, and to mirtazapine. An important finding. So if you were using this clinically and you saw that the patient was in this group, you might think about a different treatment for them. ECT or cognitive behavior therapy instead of, say, antidepressant treatment. So in the last minute I want to talk about inflammation, because this is a cutting edge psychiatry. We're very involved in this research. You all know that inflammation is important in cardiovascular disease, diabetes, cancer. But there's increasing evidence that it has a role in depression. There's an increase in inflammatory cytokines in a subgroup of patients with depression, and these patients do more poorly with conventional antidepressants. And this increase in inflammation occurs both in blood and in cerebrospinal fluid. And so Andy Miller, one of my former colleagues, and Chuck Raison did a study with infliximab, which is a TNF-alpha antagonist, and I just want to show you the data. So this is a drug that's given by intravenous infusion. They looked at 60 patients, 30 per group, and they gave them 1 to 3 infusions over a 12-week period. And these were patients who had failed 1 SSRI trial. Typical depressed population is a small pilot study, mean age of 40 years, mostly women. They had a high unemployment rate, 40%, highly educated group, and the results look terrible. Infliximab did not separate from placebo. This is a failed trial. Now, why didn't this work? It didn't work because the patients weren't selected to have inflammation. These were unselected patients. But if you then looked at CRP levels at baseline and looked at treatment response, you can see that the patients with evidence of inflammation, defined by the American Heart Association of a CRP greater than 3, you could see they had an excellent response to this TNF-alpha antagonist. And now you can see a nice effect of the drug compared to placebo, only in the patients who had elevations in CRP levels. So I would suggest to you that there is a sweet spot of inflammation. Inflammation is there for a reason, and it's important to not reduce inflammation. And the patients who received the TNF-alpha antagonist, who had low levels of inflammation at baseline, actually got worse after treatment with TNF-alpha. This is the heart of personalized medicine. And we've just designed a study using mesenchymal stem cells, which is the treatment of choice to reduce inflammatory cytokines across the board, TNF-alpha, IL-6, IL-1. We're looking at treating refractory depression. So we have to get away from the old model of monoamine associated depressions. We have to get away from that. It's not served the field well. We're not as effective in treating depression as we think we are. We need to do better. We need to begin to look at predictors of response to psychotherapy, evidence-based psychotherapy, as well as pharmacotherapy. We need to give up the old models and begin to look at the new genetics and epigenetics. And by understanding these diseases, we'll have a better idea of how to manage them. So I'm going to stop here. Thank you.